In order to understand how neurons function, we have to remember that neurons are excitable cells. That is, they change their membrane potential in response to stimuli. The resting membrane potential of a neuron is about negative 70 millivolts. That means that a neuron is more negative on the inside and more positive on the outside, based on the concentration of negative and positive ions on each side of the membrane. Because there's a difference between the inside and the outside of the membrane, we say that the membrane is polarized. And let's do a quick review of the factors that are involved in setting up this membrane potential, this polarized difference between the inside and the outside of the cell. The first factor is the sodium-potassium ATPase pump. Remember that that's going to pump sodium out of the cell, 3 sodiums out, and potassium into the cell. 2 potassiums come in. As we keep moving 3 positive charges out and only bringing 2 positive charges back in, we're setting up a situation where the outside has more positive charges than the inside. Second, the membranes of neurons have leaky potassium channels, channels that let potassium leak out of the cell. Since we have a high concentration of potassium inside the cell, and whenever those channels are open, potassium will leak out, and that's going to move more positive charges to the outside of the cell and fewer on the inside. The final important component is that the inside of the cell contains a number of large, negatively charged molecules. They're too big to get out of the cell, and they contribute to the inside of the cell being more negative than the outside. When the neuron receives some sort of a stimulus, it changes its membrane potential by opening ion channels. Opening ion channels lets ions go in or out of the cell. That changes the positive and negative balance, and it changes the membrane potential of the cell. Neurons have ion channels that can respond to a lot of different things. Depending on what the neuron is able to detect, it may have ion channels that respond to chemicals binding to them. It may have ion channels that respond to changes in temperature. There may be ion channels responding to changes in pH, or ion channels responding to physical tension. All of those potential stimuli could open the ion channels of a particular neuron in order to change its membrane potential. When we're considering the sort of effect the stimulus can have on the membrane potential, we see that there are 2 main types of stimuli. An excitatory stimulus is a stimulus that results in opening sodium channels. When we open sodium channels, sodium rushes into the cell and makes the inside more positive. This is what's called a depolarization. Since we did have a big difference between the inside and the outside, but when we bring the positives inside, we have less of a difference as the inside becomes more positive. That's depolarization. Depolarization makes the neuron more likely to activate a signal going to the next cell. That's why we call it an excitatory potential. It's exciting the neuron to send the signal to the next cell. Some stimuli are inhibitory. Inhibitory stimuli open potassium channels in the neuron membrane. When we open potassium channels, potassium will rush out of the cell. That's more positives moving to the outside and fewer positives inside. That makes the inside of the cell even more negative than it was before. We call that a hyperpolarization. It's getting even more different between the inside and the outside. Hyperpolarization makes the neuron less likely to send the signal to the next cell, which is why we call it an inhibitory stimulus. These excitatory and inhibitory stimuli that are opening channels and causing changes in the membrane potential create what we call local or graded potentials. A local or graded potential is a small change in the membrane potential, just a small change in the membrane potential caused by the stimulus. Local potentials have a couple important properties. First of all, they are graded, which means a stronger stimulus can cause a stronger change in the membrane potential, and a smaller stimulus will cause a smaller change in the membrane potential. These graded potentials are referred to as being local because we have the strongest effect right at the site of the stimulus, but then as you get further from the stimulus, there's a decrease in the amount of membrane potential change. So we get a membrane potential change right where the stimulus occurred, right on that particular dendrite, but we don't have much change further away. If we take a look at this drawing of a neuron, you can see the neuron with its dendrites on this end, and the dendrites are receiving several different stimuli. We have a stimulus coming from X, from Y, and from Z. Notice that when stimulus X occurs, the membrane potential rises a little bit. It depolarizes. So we can say that stimulus X is an excitatory stimulus, and it's going to cause an excitatory potential, a depolarization. Stimulus Z, on the other hand, when we release stimulus Z, you can see that our membrane potential actually goes down. We decrease the membrane potential, causing a hyperpolarization. This is going to be an inhibitory potential. Another interesting property of these graded potentials is that they add up. We can sum them together in order to get a sort of cumulative effect. For example, if I have stimulus X, and then I immediately have stimulus X again really quickly, I get a change in membrane potential from the first time we got a stimulus at X, and I get additional change in membrane potential from the second time we stimulated X. So we've increased the change in the membrane potential because these potentials can add up over time if they're close enough together. You can see that we could add X and Y together. This is referred to as temporal summation, where having signals close together causes them to add up to a bigger membrane potential change. We can also see what's called spatial summation, where having two different stimuli at the same time adds them together. So we get sort of a cumulative effect of different stimuli. For example, if we take stimulus X, which is a little bit excitatory, and stimulus Z, which is a little bit inhibitory, and both of those are signaling at the same time, they cancel each other out and we end up with no overall change in the membrane potential. If we take a look at stimulus Y, which is a stronger excitatory potential, and we pair that with stimulus Z is going to reduce the overall effect of Y. So we get a little bit of excitation, but not as much as we would if we wouldn't have had those combined together. If we combine X and Y together, we're going to get a stronger excitatory potential because they both are going to be exciting the membrane potential. They're both going to cause depolarization. This sort of summation is happening within a neuron all the time. All the various signals that are gathered from all the different dendrites are summed up at the axon hillock. So this is where we're really measuring where all these graded potentials are coming together. They're all summed up at the axon hillock. And this is what's going to determine whether the signal is going to be sent on to the next cell or not. If the depolarization is strong enough, if we raise the membrane potential enough, we might trigger an action potential. The threshold depolarization for triggering an action potential is around minus 55 millivolts. I think of this as the magic minus 55. If we get graded potentials up to magic minus 55, that's where we trigger an action potential. We don't just go up and come back down, we have something special that happens. An action potential is a very large depolarization. We're talking about a depolarization that goes all the way up past 0 to positive 30 millivolts, a big depolarization. And an action potential is all or none. We either trigger a whole action potential or we don't trigger an action potential at all. If we get to that magic number, we're definitely getting an action potential. And you can't have a stronger or a weaker action potential because it's all or none. You either get an action potential or you don't. Action potentials are always a depolarization, and action potentials spread along the axon. This is what's actually going to carry the signal to the next cell. When we trigger an action potential, big depolarization that spreads along the axon and will trigger the release of the signal to the next cell. The reason we need to look at the axon hillock, or that initial segment of the axon, is because that first part of the axon has a lot of voltage-gated channels. As long as the membrane potential remains below that magic minus 55, those voltage-gated channels are closed. As soon as all the sums of all the graded potentials reaches up to the minus 55, It opens those voltage-gated channels at the beginning of the axon, and that's what leads to the action potential. We saw the same phenomenon when we were looking at the action potential in a muscle fiber. Once we opened voltage-gated channels, ions rushed in and changed the membrane potential, which opened more voltage-gated channels, allowing more ions in, opening more channels, letting more ions in, and that is what spreads along the entire axon to carry the signal to the next cell. Let's take a closer look at the parts or the phases of an action potential so we get an idea of what's happening at each of these parts of an action potential. If I were to electrically measure the change in the membrane potential of an axon as an action potential goes through, it looks like this graph. We start at rest, at the resting membrane potential, then we have a rapid depolarization that goes up very high. And then we have a repolarization as it comes back down. And there's a little hyperpolarization where it goes a little bit too far, down past the normal resting membrane potential. And then we come back up to resting again. At rest, those voltage-gated channels are closed. When we're looking at an action potential, we have to consider both voltage-gated sodium channels and voltage-gated potassium channels because they're both at work here. And both of these are closed. So potassium's not going out and sodium isn't coming in. Once we reach the magic minus 55, that's the trigger that opens these channels. Both the sodium channels and the potassium channels are triggered to open at the same time. But the voltage-gated sodium channels open fast and the voltage-gated potassium channels open slowly. So for a while, after we reach that magic minus 55, we've had those sodium channels spring open and sodium is rushing into the cell. As sodium rushes into the cell, the inside of the cell becomes more positive. That's why we see this increasing depolarization of the action potential as the membrane potential gets higher and higher. This depolarization is going to open nearby sodium channels, which will let more sodium in, which will change the membrane potential in order to open other channels, which lets more ions in. This is going to spread all the way along the axon. Notice that the sodium channel has a structure called an inactivation gate. This inactivation gate closes off or blocks the sodium channel. So at this point, we've blocked our sodium channel. No more sodium can come in. About this same time, the potassium channels that started opening as soon as we hit magic minus 55 finally finish opening, and potassium begins rushing out of the cell. As potassium with its positive charges rushes out, we're losing positive on the inside, and the membrane potential begins to come back down as the potassium rushes out. Once we get back to that minus 55 again, that's where the sodium channels are going to be closed, so we don't have to worry about the inactivation gate anymore because the channels are closed again. And our potassium channels are beginning to close, but remember that they're slow. So while they're in the process of slowly trying to close, more potassium is rushing out, and that causes a hyperpolarization. Our neuron actually gets an even more negative membrane potential than it has in its resting state. Once our potassium channels finally get closed, then our sodium-potassium pumps can start resetting the membrane potential back to its resting state. At this point, we can trigger another action potential and start the series of steps all over again. This GIF animates the way the action potential moves down the axon. So at any one point, we're letting sodium into the cell, which will then open the sodium channels in the next segment, changing the membrane potential there, which then open the sodium channels in the next segment and open the sodium channels there. At the same time, we're resetting the membrane potential behind that change. So while one section of the axon is depolarizing because we've opened sodium channels and sodium is rushing in, the section just behind it has already inactivated the sodium and is letting the potassium out in order to reset the membrane potential there. So this is a wave of depolarization that spreads along the axon. The neuron that's shown in this GIF is an unmyelinated neuron. So we're going to see a real steady process of that action potential along the axon. If we were looking at a myelinated neuron, this change in membrane potential would hop from node to node in saltatory conduction to give us a faster action potential. I mentioned before that action potentials are all or none. We can't sum up action potentials. I can't start an action potential and immediately start a second one to get a super strong action potential. And that has to do with the refractory period. There's a refractory period associated with an action potential during which you simply cannot start a new action potential. Let's think about why that may be. When we start an action potential, we've opened all of the sodium channels in that area. They're all open and the sodium is rushing in. So you could try stimulating again, but the sodium channels are already open. You're not going to get an action potential because you already have one in progress. If we wait a little bit longer until we get to where we're repolarizing, the sodium channels are inactivated. They're open, but they're inactivated, and it doesn't matter how much signal you give them, how strongly you try to do anything with them, no more sodium can come in because the gates have been blocked by the inactivation gates. So you can't change the membrane potential with those sodium channels. You can't get any more depolarization. Once we get back down into the hyperpolarization phase, our sodium channels are closed again and those inactivation gates aren't a problem. So we can reopen our sodium channels, but keep in mind that the membrane is now hyperpolarized. It's going to be even harder to get enough graded potentials to add up to magic minus 55 than it was before. During the depolarization and repolarization phases of the action potential, we call that the absolute refractory period. There is no way to start another action potential no matter how hard you try because the sodium channels are not going to be able to respond. Once we get into the hyperpolarization phase, we can open the sodium channels and start an action potential, but it's much harder. This is called the relative refractory period because you can start a new action potential, but you have to have a lot of graded potentials to get back up to minus 55 from that hyperpolarized state.